Our research is mainly focused on cartilage. As you probably know, cartilage is the tissue that lines the articulating bones in our synovial joints. It's not only present in joints, of course, it's also present in the airways, in the larynx, in the nose, and in the ear. Cartilage is a very specific and very interesting tissue as well. It is made up of chondrocytes, and these chondrocytes are embedded in a peculiar matrix, and this matrix is responsible for the functions of cartilage. So it resists compression and absorbs mechanical shocks during joint function. But of course, just like everything in our body, some diseases, some conditions can affect cartilage as well. I'm sure that most of you have got relatives, friends, or grandparents or parents who are suffering from joint pain. And you probably know that this is a very debilitating condition. And unfortunately, other than giving the patients painkillers, there is only so much that we can do about it. One of the major contributors to joint pain is osteoarthritis. During osteoarthritis, articular cartilage gets damaged, it gets degraded, and the main features of this condition, like I said, is joint pain and joint stiffness. We have historically considered osteoarthritis as a kind of wear and tear disease, and we thought that it's part of natural aging. So this means that as we grow older, our cartilage gets thinner and weaker, and we tend to have OA. But this is not entirely the case. As you can see on this figure here, now we consider osteoarthritis as not only the disease of articular cartilage, but also the entire joint, so it's a whole joint disease. And it's not a single condition as well. There are, as research progresses in this area, there are multiple forms, multiple phenotypes, and there are different molecular pathways that lead to similar symptoms in patients. But like I said, even to date, there is only so much we can do with the patient. So there are some emerging therapies, there are mesenchymal stem cell-based therapies, and there are other biological therapies as well, but none of these have proven to be effective in all patients. So this condition represents a significant unmet medical need, and that's why we are trying to look at the mechanisms of this disease. Studying articular cartilage, especially its formation, is kind of challenging because this tissue forms prenatally, so in utero when we are embryos and fetuses. So studying the normal formation of cartilage in humans is not easy. But we hypothesized that if we better understand how joint tissues, especially articular cartilage, forms in utero, so in the embryo and in the fetus, hopefully we will be at a better position to develop novel or more efficient approaches to help patients suffering from this disorder. There are different ways of studying tissues. You can choose to work on specific targets, like receptors or other proteins, or you can look at the bulk protein or mRNA expression, and the latter case is known as the omics approach. We can look at the tissues or cells in interest at different levels. So you can look at, for example, the DNA content. This is known as genomics. You can check whether there are any mutations or chromosomal abnormalities associated with the disease. Or you can look at the epigenome. This is epigenetics. There are, as you know, or probably know, there are modifications on certain DNA sequences or histone proteins, and this is going to affect DNA or gene expression. Then you can look at the transcriptome, so that you can have information of all the messenger RNAs and other types of RNA molecules that are expressed in a tissue at a given time. This is known as transcriptomics. And there are different levels to it. I'm going to show this on the next slide. And then, in the next level, you can look at the proteome as well. These are all the proteins that are expressed and present and probably functional at the cell or tissue at a given time. And then you can go even further. You look at the metabolomics as well, all those metabolites that the proteins produce. I'd like to focus on transcriptomics. And this is a slide that summarizes the different levels of how you can look at the transcriptome at the global scale. You can do microarrays. This is a bit outdated these days. You can do bulk RNA sequencing, which means that you have a tissue with many cells, and you isolate or extract RNA from all those cells at the same time and look at the global mRNA expression pattern. This is what we did. But now you can go even further details as well. So now it is possible to gather information from a single cell, which is cool, I think. Or even you can go into organelle level, and you can look at the mRNA expression patterns in a single nucleus. Or you can do spatial transcriptomics and multi-omics as well. So if you take a slide, for example, a joint in this case, you can look at mRNA expression or even protein expression, spatial expression patterns, so whether it's in cartilage or bone or synovium or other tissues. What is chondrogenesis, and why do we care about this? So chondrogenesis, the formation of cartilage, starts off with mesenchyme. This is also known as embryonic connective tissue. As you can see, mesenchymal cells are kind of elongated or spinous with a fibroblastic appearance. Fibroblast is a basic connective tissue cell type. And as you can see, the cells are loosely arranged. So this doesn't resemble a cartilage, right? But as a result of certain secreted factors and intracellular proteins and their interactions, something starts to happen with these cells. They proliferate, so they increase in number, and they get closer to one another, forming little nodules. And these chondrogenic nodules are essential for differentiation, which means that these cells lose their original morphology and they become rounded or oval-shaped, so they acquire this typical chondrocyte, cartilage cell morphology, and they also start producing cartilage matrix. And like I said, this process is difficult to study in humans because it happens in the embryo, so there are different models, experimental models, that we can study the process. So what we set out to do is that we wanted to gather information on the transcriptome, on the RNA expression patterns and landscape of a chondrogenic model, because we thought that if we understand, if we really understand what mRNA expression patterns characterize chondrogenesis, they may find some interesting targets that could be targeted in patients, or just to enhance chondrogenic differentiation. So what we did, like I said, is the transcriptome analysis. So the transcriptome is the full range of mRNAs expressed by an organism or a tissue or a cell type. And we were focusing on transcription factors as well, because it's not enough to know what mRNAs are present, but we also need to know how the expression of these molecules is regulated. And transcription factors are those proteins that are necessary for the RNA polymerase to bind to specific regions of the genes and start genes transcription. So this is the model that we used. It seems interesting, right, that we are using chicken embryos, but there is a reason for that. Eggs are really easy to handle, and you just pop them into the incubator for 4 days, and then little chicken embryos will form. And this model is very good at recapitulating human cartilage formation as well. So we isolated these chondroprogenitor cells for the developing limbs of these chick embryos, and we established so-called micromass cultures, which means that we inoculate the cells in a little droplet, and this is going to mimic condensation and nodule formation. And then these cells, if you treat them in just normal tissue culture media and in a carbon dioxide incubator, these cells will then fulfill their differentiation program and form cartilage tissue in the lab. We can stain these cartilage tissues with dimethylmethylene blue, and as you can see, it's blue, but the staining is like purple because this is the metachromatic dye. It's going to show cartilage matrix formation. And so then we isolated RNA from cultures on different days, we RNA sequenced, and then we analyzed the data. Okay, so we've got all the mRNAs extracted from the cells, so let's see what we see. And we use computer analysis to look at for example, the trajectory or the variability between samples. We did a so-called principal component analysis, and this is a very good method to kind of scale down the variations in this very highly complex data set. And as you can see, we also did some so-called trajectory analysis, and both indicate that there is a progress, right? There is a trajectory that these cultures kind of follow during differentiation. So we could confirm what we see using conventional techniques. We could confirm this at the mRNA level that, yes, there is a differentiation taking place. Okay, so now we have the expression values as well of all the mRNAs that are present in the tissue. So we generated heat maps. A heat map looks like this. It's very interesting. So each column represents a sample, and each little box represents an mRNA, and the colors are assigned depending on their expression levels. So red means high expression, and blue means low expression. You can cluster these, and you can infer some interesting expression patterns, like there are some genes that increase over time, there are some genes that decrease over time. We can do a lot with this data. I don't want to go into details. So we were looking at transcription factors specifically with the hope that if we target these transcription factors that have not yet been implicated in chondrogenesis, we can maybe generate better quality cartilage for patients to implant, for example. And we picked these two, 808 and EBF1. None of these have been previously described as important regulators of chondrogenesis. So we did some analyses as well, and we confirmed their expression and regulatory role in chondrogenesis. So if we silenced any of these, then chondrogenesis was going down. But of course, the data set is there, so we are going to continue to look at these data, and we will hopefully identify some other regulatory targets as well. So in summary, in this work, we have described the transcriptomic landscape in this dynamic chondrogenic model, which brought us a bit closer to understand how cartilage forms. And we identified these two transcription factors that have not yet been implicated in chondrogenesis. And like I said, a deeper understanding of the molecular regulation of cartilage formation by these transcription factors and the downstream signaling that is associated with them could be exploited for regenerative medicine. Because osteoarthritis is a progressive disorder. Sometimes it takes decades for a patient to develop this condition, and even before the symptoms arise, there is a so-called molecular phase when the changes are already happening, but pain or joint stiffness is not yet there. So there are windows of opportunities, and hopefully if we interfere in time, we can maybe save patients from suffering from this disorder, or at least to delay the onset of osteoarthritis. And with that, I would like to thank to the team that were involved in this work, especially Judith and Roland. They were the first authors of the paper, and to everyone listed on this slide, and thank you very much for the attention.